Pavitra, who have been part of IVAST and part of Beacon for a long, long time. And they're going to present some of the long-term work that they've been doing, combining mathematics and wet-bench experiments and experimental evolution in spatially constrained environments. And with that, I'll turn it over to Steve. Yep. Okay.
Hi, everyone. So I'm going to be starting the first part of the talk. That'll be the warm-up act for my PhD student, Pavitra Roychoudhury, and she'll bring it home from there, hopefully about the halfway point. So what I want to talk about today is spatial evolution and ecology, and we're going to focus on pathogens here, mostly bacteriophage. A lot of emphasis hold true, I think, for general pathogens. And we're going to look at these things from the point of view of mathematical models and experiments. But I'm a mathematician, and so I'd like to slant the discussion just a little bit towards the theoretical side. We'll see plenty of experiments as well. So what's driven a lot of my research over the years is interest in the effects of spatial structure. So all biological communities in nature are spatially structured in one form or another. It's really hard to avoid spatial structure. So the question that drives a lot of my work is how does spatial structure influence phenotypic changes and evolutionary trajectories during adaptation to a novel environment? So what we're going to talk about here is mostly microbial experimental evolution on the experimental side, and modeling. So in the experiments that I'm going to talk about, we'll be looking at phage bacteria systems. And the mathematical models are individual-based, they're stochastic, and they're explicitly spatial. So these models go under various names. I like to call them interactive particle systems. Some people call them cellular automata, CA models, and there are a number of other terms as well. So I mentioned spatial structure, and this is well for microbial, the importance of spatial structure. And in particular, these organisms have evolved in spatial environments, and so that should leave some signature on how they evolved, what phenotypes of their evolutionary trajectories look like. Some of the other things that I like to inject into the work is something that's different from experimental systems often is that in the real world, environmental changes are happening all the time. These might be biotic or abiotic changes. And these changes in environmental conditions can be quite strong. They can alter selective pressure, and in particular, they can lead to what I call spatial bottlenecks. So this is another thing that I think is missing from experiments in theory, or at least underrepresented. Biological communities go through periods of boom and bust. So there are periods of decline when the environment is not so good, and then subsequent expansion.
Steve, could I interrupt you for just a second? The overflow room is online, so anyone who would like a place to sit or a more comfortable place upstairs, the classroom on the fourth floor is ready to go. Feel free. If you want to remain in the presence of the stage.
So what Pavitra and I are going to do is go through 3 examples. And these are going to give some partial snapshots of some of the work that has been going on in my group and is going on right now. I'm going to cover the first 2 examples. Sorry, in order of increasing complexity. And the very first system is going to be just a theoretical model. And we're going to look at what happens when we have a host pathogen system, the pathogen is evolving. What effects are there of spatial structure in, say, the evolution of virulence? In the second example, this is a joint theoretical experimental project that I colleague recommends. And we have competition between 2 phage species, common bacterial hosts. And what we did is we grew these on surfaces on moderate plates. And we monitored the effects of various things on the competition And then finally, Pavitra is going to take over the ASLE with a description of spatial adaptive evolution of a single phage. We're going to be looking at optimal phenotypes. Again, this is both theory and experiment. Okay, so before I start the example, let me just say a few words about the mathematical model. So I'm going to call it an interacting particle system. So here's a little picture of our cartoon of the spatial grid that we're talking about. So keep that in mind as I tell you about these features. So we have this grid of sites representing discrete spatial structures. At each site, you can have different states. So a site might be vacant. It might be infected by bacterial cells. Those cells might be infected. They might be uninfected. There might be different species. They might be resistant to the virus. We'll have 3 virus cells as well. The models include randomness and spatial structure down to any level you want. Typically, we have to go down to the individual cell level. You can also go down to the level of individual virus particles. In the models, we build in various life history parameters that we think are important and we want to pursue. For example, a phage, a virus, we might include the birth size, latent period, lysis time, absorption rates. For the bacterial host, we can put in growth rates, resistance to various phages, and so on. We can also incorporate co-infection dynamics and recombination. And that might be relevant under the high multiplicity infection conditions that we see on the plate. Okay, so when I show you some simulations, you're going to see different colors on the grid. They're going to be moving around, doing various things. Changes in the colors are just the different changes that are coming through the model. So I'm not exactly explaining what the model is doing yet. I'm going to let that unfold as we go. Okay, so here's the first simple example. So a lot of you know about classical, the simplest models of epidemiology, the SIR model. So susceptible, infectious, and group categories for our population. And so what we typically learn about first is a mass action ordinary differential equation. And so here you see the rates of change for the susceptible population and the infected population. We have an infection rate beta, so we only lose susceptibles in this model due to infection. And there's this mass action interaction term, which is just given by the product of 2 global densities. And then the infectious population grows through infection, and infectious individuals are lost through death. Okay, so what we wanted to do here is try to understand, if you're thinking about something like the evolution of virulence, what types of mutants would be likely to arise in a given wild-type population? What properties, what infection rates and death rates would allow them the population? So now what we're going to do is think about 2 types of pathogens. So infection rates beta 1. 1 is the wild type, beta 2 for the mutant, and then death rates delta 1 and delta 2. Okay, so in this SIR model, a lot of the theory is governed by the basic reproductive ratio. So in order for the epidemic to spread, we need this quantity R0 to be greater than 1. Now, if we put 2 pathogens in this well-mixed environment, let's assume that the host is susceptible to both of them, then both pathogens see the same density of host. And so, basically, the winner is determined by the ratio of infectivity to virulence, death rate. And so, whoever has the highest ratio is the winner. So, any combination of the change in beta and delta that gives you a higher ratio leads to invasion. So now what happens in a spatial environment? So now things aren't well mixed. Now different infectious individuals do not see the same density of hosts. So what we did in this project, which was actually driven by a question from Jim Gold, expanding wave of an epidemic or a virus. So this might be a flat, a phage flat, for example. We're going to inoculate some host population with a pathogen, a phage, and it's going to start expanding radially. And we're going to let mutants arise spontaneously in different properties. And one of the things that Wolf pointed out early on is that when you're looking for successful mutants that occur back inside the wave where there are all these dead hosts, they don't have many susceptible hosts to grow on. And so they're not likely to make a difference. So all the action is on the wave front. Well, let's go now to a simulation. So what you're seeing here is the green represents susceptible hosts, or waiting community. The red are the actively infected hosts, and the yellow are the dead hosts. So we're talking about phage, the yellow would be the lysis cells. And as this wave of infection moves through the population of susceptibles, we see that there are some different colors right here. So the pathogens are colored black when they're actively infected. Kill the host, the color turns to blue. But what you see here are clonal sectors of various mutants that are appearing on the edge. And sometimes what you'll see in simulations is that a mutant that appears sort of toward the inside of the wave often doesn't have a chance to succeed. So there's a whole lot you can do to describe diffusivity and things like that, what role that has to play. But basically, we built this model to try to understand in the spatial landscape what can change the infectivity and the death rates. What values of those likely lead to a successful invasion? So something that might evolve. So one of the things that we did with this is we just ran a bunch of simulations. We had a wild type pattern with a given delta and beta. And then we varied over a large space different values of the mutant infectivity and the mutant death rate. And we asked when was the probability of invasion greater than a certain threshold. So that's the successful ones are the part that's given by this slope surface here. And if you simply fit various curves in data, what you see is that what matters is not the ratio of beta to delta. It's actually it's that ratio times the first power of the infectivity. So infectivity is much more important in determining success in this simple model. So this model doesn't have anything like co-infection or other properties like that. It's very simple, but it suggests something. That first of all, there are different rules governing competition and evolution in a spatial setting, and that perhaps what really matters is your spreading ability, your ability to get fresh hosts, at least in expanding ways. In the second example, now we're going to get a little bit more complicated. So this was an experimental system that Holly developed, where we serially transferred on auger plates two different kinds of phage. So it was 5x174 and alpha 3, ovolytic phages, and they have a common host, E. coli. So these were grown on these plates for a certain incubation period. And then after a few hours, the phage basically cleared the lawn of host cells. And you have to keep the evolution going, you have to transfer to fresh host cells. So we use a replicate picker, a bed of nails, to transfer a sample of the phage to the next lawn of host cells so that we could continue. We kill all the host cells so that we're not transferring things like resistant cells. Okay, so what happens if you first start looking at this, what you quickly see is that somehow propagates better in spatial environment, in this spatial environment than alpha-3 does. So you'd expect it to win a competition. And so what we did is we tried a situation. We ran a competition in what Holly likes to describe as a civil war scenario. So we put phi x on the top half. We inoculated the cells with phi x in the top half of the plate, alpha-3 in the bottom half of the plate, and we just let them go at it. And in the short incubation period, 5 hours, the plate, phi x wins. What we expected. So this top row here is that initial experiment. Short incubation period, and what you see, 15 passages of this experiment. Here's the initial Civil War startup. Yellow is phi x, blue is alpha-3 only, and then the green region is a sort of a hybrid zone where we have phage particles of both types present. So if a cell is in that green region, it can be infected by either type, and it's just a question of action speed. Who wins? And we don't allow full infection in this model either. So what you see in this first row is that alpha-3 takes, or pi x takes over the short incubation time. The next row is the mathematical model that we ran. I'll say a bit more about it in a minute. Now let's go down to the next row here. So in B, we increase the incubation time from 5 hours to 18 hours, long after the plate had been cleared of most of the cells, resistant cells. And surprisingly, we were surprised, we saw that phi x and alpha-3 both coexist. The weak competitor was able to persist somehow in this setting. And so early in the game, we had discussions trying to figure out what was going on. Was alpha-3 evolving? There's some kind of arms race going on. And so we decided to build some mathematical models to try to go through various scenarios, try to understand what was going on. And so one of the simplest explanations is that this might have to do with resistant cells. So you see resistant cells popping up all the time in these plate experiments. And so one of the differences between the long incubation and short incubation experiments is the length of time that resistant cells have to grow. So we built that into the model. In the short incubation period, I'll show you some simulations. The resistant cells didn't have time to spread very far. In the long incubation periods, in these clusters of black that we see right here, those are colonies of resistant cells that have spread and run into the green region that contains alpha-3 cells. There are some differences between the experimental results and the simulation. So this hybrid zone is a lot thicker in the experiments than in the simulation. And that has to do with the genotyping that we did. I'll try to say more about that. Okay, so let's go to simulation. See how the experiment ran. Okay, so here's the start of the experiment. So there's the Civil War scenario. The green represents basically nutrient, empty space. There's a magenta color, I guess. That's most of the host cells at a fairly low density in this picture. The yellow, here you see a circular yellow patch. That's a single burst of a cell that was infected by phi x. So maybe about 100 progeny was spilled out and diffused a little bit. The yellow represent free phage. The red, you can see them within these things, are cells that are infected by the yellow virus, the phi x. Down in the bottom, the blue represents alpha-3, the weak competitor, free phage, and there are little black dots in there that represent the infected cells. Now we run this, we let the plate clear, so all the magenta would leave. You see a hybrid zone developing, where both types of phage present. And then let's say that's 5 hours. So what we do at that point, we kill all of those cells now. We use use our replicate picker to sample the phage, and then we transfer them to a lawn of fresh host cells, and we start the next incubation period, 5 hours again. And I'll let this run for a bit. And what you'll see is that phi X, the yellow, starts pushing down, starts to take over. Even though alpha 3 is still infecting things, phi X starts to win. And it only takes about 5 to 10 incubation periods for alpha 3 to go to the. Okay, so phi X is way down here now. Alpha 3 is up here, but in time it'll be lost. The other thing that you're seeing up here are these little black splotches. And I've artificially increased their rate of appearance and spread so you can see them in this short talk. These are the cells that are resistant to phi X. So they appeared spontaneously, and they're able to grow and spread in the phi X dominated region. These are resistant only to phi X. Alpha 3 can infect these cells. There are also cells that are resistant only to alpha 3 and cells that are resistant to both. Turns out they don't matter. Okay, so in the short incubation periods, these resistant patches of cells that are serve as a nice refuge for alpha 3 aren't quite large enough to form percolating clusters that would help survive. So now let's restart. Now I'm going to let it run longer. You guess what's going to happen. So we let the competition unfold, and maybe that's about 5 hours right there. The plate is pretty much cleared. And now we just let it sit in the incubator overnight. And the clusters of resistant cells they have already appeared, and they appeared before, but now they can start spreading. And what you'll see happening is that this lower cluster of resistant cells is going to grow into the hybrid zone, which already contains alpha 3, the weak competitor. Once it hits that, alpha 3 will start infecting those and will spread in a wave of advance up into that cluster. You'll start seeing blue appearing up there. There it goes. Okay, so it starts spreading. So the parameters I have in this model are a little bit off, just so you can see it playing right here. But you can guess now what's going to happen. These things are going to start spreading. They have this spatiotemporal refuge of the resistant cells. Back to this. A couple of the things that we saw in experiments, I just want to point out some of the things that didn't match between the theory and the experiment. So in the top row here, this is the case of short incubation. Here's the experiment on the left. Alpha 3 dies out, phi X takes over the whole place. Here's the simulation. Same thing happened, but the oscillations are a little bit smaller. It's even more dramatic in the bottom row, where we have the long incubation periods. Again, phi X takes over, but alpha 3 persists now in the experiment. Same thing happens in the simulation. They match quite well, except there are these large fluctuations in the experiment that weren't in the simulation. So this happens for several reasons. But one of the things that we can do with simulations is, with our model, is we can see with absolute clarity what's going on our plate. We can count the exact number of each type of bacterial cell in the phage. And in the experiments, we're limited. Our ability to count cells in the phage of different type are very limited. And so we have to use different methods to do it. So here we counted the presence of the two different types of phage by plating them, using the replicate picker, and then plating them on selected host cells that showed what's going on. And in the simulations, we just counted exactly what there was. But if you mimic the experimental conditions, so here's what happened there with the exact counting. If we go to the exact, sort of the replicate picker genotyping we develop these oscillations that we were seeing in the experiments. And if we make this genotyping even more clunky, so we separate the replicate from the bigger prongs even more and make them bigger, we enhance the fluctuations even more. So here's just a simple example of a case where it looks as though the simulations are not matching certain features of the experiment. Instead, what they're doing is pointing out artifacts of the experimental protocol. So I think it's time to move on to Pavitra and the second and third example.
Right. Hi everyone, my name is Pavitra Roychoudhury and I'm a PhD student here at UNI. And I'm going to be talking about some of the things that was related to here. When I came to the UI in 2009, the lab was in the middle of a series of evolution experiments where we were trying to understand the effects of spatial structure on adaptive evolution in response to a thermal stress using the bacteriophage ID. So very briefly, what the experiment was doing was to inoculate a lawn of bacteria with wild-type ID11 and passage this onto fresh hosts at the end of every incubation period in order to observe hundreds of generations of phage evolution. So one of the things that I thought was really cool about microbial systems like bacteriophage was its ability to map genotype to phenotype to fitness, which is something that's quite difficult to do in other organisms, and I thought that was really cool. So I realized that here I have access to this nice library of evolved phage that I could then compare in terms of both genotype and phenotype to the ancestral population and also measure its fitness. And so since the ID11, the bacteriophage that's pretty small, has a pretty small genome, you can actually realistically do whole genome sequencing of the evolved phage. Phenotypes are fairly easy to measure, and the fitness of the bacteriophage is dependent on just three primary phage traits, which is absorption rate, lifetime, and burst size. And in addition to traditional measures of fitness, like the population growth in terms of doublings per hour, we can also measure spatial measures of fitness, like class size, which is how big is the clearing that this phage produces when it's growing in a lawn of bacteria, and plaque density. If I stab this growing plaque in the middle and using and measure how many phage are in a small area, how many are there, that gives us another measure of spatial fitness. So the way I like to think about this is using this metaphor of fitness landscape, and I particularly like this cartoon that I stole from the Beacon website. So this is an example of a fitness landscape, and I think this is something that everyone is familiar with, but very briefly, populations occupy some position on this fitness landscape, which is basically plotting fitness against either phenotypic or genotypic space. And mutation and recombination will push populations away from their current position, and natural selection will drive populations up these slopes of increasing fitness. And other processes like drift and bottlenecking, for example, will push the populations back down or up. So what is the effect of spatial structure then on adaptive evolution? So spatial structure, conditions of spatial structure allow populations to explore multiple adaptive trajectories. And this is because in a spatially structured environment, competition becomes localized. So a beneficial mutation of small effect can spread in a smaller spatial region and persist for longer than in a well-mixed environment, which is typically subjected to big selective sweeps. So what we expect to see is basically, first of all, an increase in diversity, and also we expect to see populations exploring these multiple adaptive trajectories. So some of the questions that we can ask are, well, what does this fitness landscape look like? Are there any phenotypic optics destination? Does it help these populations reach these optimum faster? What about the role of bottlenecks? How does that impact evolutionary trajectory? And what I was really interested in is, well, can we make predictions about any of these processes using mathematical models? So what we did was we developed this mathematical model, or interactive particle system, which Steve talked about. And this was inspired by these experiments that were going on at the time. And so very briefly, we have a two-dimensional grid of sites, and each site can contain a cell and phage. So this is the difference between this model and some of the earlier models that we saw, which is that we also track the states and populations of phage at each site. So an uninfected cell at a site that contains phage can become infected at some rate that depends on the absorption rate of the phage that are at that site. And then that infected cell then goes on to lyse after a length of time that depends on the latent period of the phage that's infecting it. And that site that was occupied by the lyse cell is then, it then becomes vacant and can be populated when an uninfected cell in the neighborhood divides. So in addition to the state of cells, we also look at the states of phage at that site. So phage are either free, which means that they haven't infected the cell yet, or they're replicating, which means they've already infected the cell and they're in the process of undergoing replication. The other thing that we implemented in the model was mutation. And here we look at mutations that affect our phenotype of interest. So the wild type phage can mutate and take on a phenotype, a new phenotype, which has a different attachment time or lysis time or a burst size from the original one. We also included things like diffusion and other phage life cycle events. So the idea was to make it as close as possible to the experimental system. The other cool thing about the model was that we can have different starting configurations and different levels of structure. So at the simple end, the simplest simulation is single plaque or epidemic simulation that you saw earlier that Steve talked about, where you just have a single starting infected cell or a single phage, and then that spreads in a roughly circular manner to form a simulated plaque. The other thing we can do is we can initiate these metapopulations in space using the version of the replicator that we talked about, and you can look at different things like the effect of the bottleneck size, spacing of the Pavitra prongs, and other things. We can also run competition simulations. So what I'm showing here on the right is a competition simulation where we basically sprinkled phage of many different phenotypes onto the grid and let the simulation go to ask who wins, what's the best phenotype in terms of competitive fitness. And since in a structured environment, your interactions are localized, by removing those localized interactions and making those interactions global, what you're doing is you're then simulating a well-mixed or liquid environment. So this is the other interesting thing, which is that the same model can be used to simulate both a structured and a well-mixed environment and ask what effect structure has. So what I'm going to tell you about is a set of simulations that were used to explain some experimental results that we found. So as I mentioned, we had this evolved population from our series of evolution experiments. And so the first thing I did when I got to the lab, besides ensuring that I don't damage anything and blow anything up, was to understand some basic phenotypic assays. So I went into the evolved population and decided to assay the evolved population to determine what are the absorption rates of phage in that population and lysis times and fitnesses. So immediately what stood out was that this evolved population of phage actually got worse at absorbing to their hosts than the wild type. So that's indicated by this pull some isolates, which we then sequenced. And what we saw was that mutation in the major capsid protein F was sufficient to significantly reduce absorption without any changes in lysis time or burst time. So what it was doing was it was making it a slower absorber with no other changes in it. So here we had another example of this genotype to phenotype to fitness mapping. And so when we went to measure spatial measures of fitness, we found that plaques that were produced by these mutants were larger and also contained more phage per unit area. So that was pretty interesting. So we wanted to know, well, can this seemingly worse pathogen in the reduced infectivity, is reduced infectivity enough to lead to higher fitness? So to explore this, what we did was we used the model and we initiated simulations with a single infected cell to produce a plaque. And we let the simulation go for 3 hours in lawns that had varying density, varying host density. And this was a very simple model, no mutation, no multiple infection. And all we did was we varied the infection probability. So varied it. So here, infection probability, low infection probability, low absorption, slow absorption, and so on. And then we basically measured fitness at the end of the simulation. And what we found was that low infectivity does indeed produce larger and denser plaques, but this result depends on host density. So this is only true when hosts are abundant. So when hosts are abundant, low infectivity maximizes plaque diameter and density. And in conditions of in the opposite scenario, when your hosts are scarce, then it's better to have rapid absorption because there's this implicit trade-off between competition for nearby hosts versus diffusing hosts that are further away. So this was the result that we got, and it was really cool because here's a situation where we were able to include something about the ecological dynamics that was not present in our original system, and ask what effect that had on our results. So that was the first thing that we did. The next question logically was, well, in this case we had latent period remaining the same, but what if latent period could also change? So what if phage could change their latent period and absorption time? So can we construct a fitness landscape? So in very similar simulations, we initiated plaques with phage that had different phenotypes. And in this case, we had 36 phenotypic combinations of attachment time and lysis time. And we measured fitness in a variety of ways. And to compare, we also did simulations in liquid to look at with the same 36 phenotypic combinations. And what we found was that if we measure fitness in terms of the rate of population growth in doublings per hour, in a liquid simulation, rapid absorption produces the highest fitness. So here, these red dots indicate the location of the optima, and it means that they're not statistically significant from each. Those points are not statistically significant from each other because the surface represents the mean of multiple simulations. In a structured environment, we found that slightly slower absorption led to the highest fitness. So this didn't seem very striking at first. And in both cases, intermediate lysis times were optimal. So then we looked at fitness in terms of our other measures, like plaque diameter and the end population at the end of the simulation. And in those results, what we saw was that rapid absorption leads to smaller plaques. And so if you're looking at plaque diameter, then it's slow absorption and rapid lysis that leads to the largest plaques. And it's pretty interesting because the cloud of points basically shift to the opposite end of the human space. And in terms of the end population, slow absorption and intermediate lysis times produce the maximum end population. So this is another example of a case where we used things from the experiments to inspire simulations that we can run to then generate new testable hypotheses. So we can actually go back in the lab and then look at the whole population and look for whether we have lysis time mutants with these optimal phenotypes. There are some other simulations that we ran as well. We also did competitive simulations and looked at optima based on these compositions. And we looked at the effect of bottlenecks in structured and well-mixed populations. And the other thing that we can do is we can also do experimental evolution in silico. So we can initiate a lawn of wild-type bacteriophage and change mutation rates or recombination rates, and then watch evolution take this population through this simulated experimental evolution setup. So that's just a brief overview of some of the simulations that we've done so far. And with that, we'll be happy to take any questions.
Any questions from offsite?
Yeah, can we ask a question from Washington?
Yeah.
As long as it's not Vancouver.
Thank you, Steve. That was really interesting. I have tons of questions, but I'll try to restrict it to one. I'm just curious, Pavitra, in the last bit that you talked about whether burst size in these simulations is co-varying with latent period, and whether you think that will have an effect in terms of how that relationship between bursts. Presumably, the longer the phage stays within the host, the bigger the burst size. So the more sort of particles for diffusing out you get. Is it right now? Are those independent? Are those co-varying with one another? And generally, do you think that's going to have an effect of lifting the latent period a little bit, or what effects do you think this will have?
So right now, burst size depends on lexicon in much the same way as your models and Jim Boll's models use it, as it's assumed to be linearly dependent on lysis time. But what we did have was this maximum. We assumed that burst size cannot increase indefinitely. The cell must run out of resources at some point. So we imposed a maximum. So what we found was increases in lysis time beyond a certain point are not going to be beneficial to fitness, because it's not increasing the burst size beyond a point, and it's delaying access to future infection opportunities as well. Does that answer the question?
Yeah, that's great. Can I squeeze one more in?
Yeah.
I wanted to earlier. Actually, this is a question for Steve. I'm just curious if you've extended the alpha 5 model to look at sort of how many different phage competitors, let's say, of different competitive abilities, can you maintain in a spatial system? Is there sort of some upper limit in terms of what you can maintain with regards to now all sorts of different resistance profiles going on at the same time, and sort of whether these things percolate with respect to, let's say, N phage competitors? Can you push it up? I expect you push it up past 2, but have you explored sort of just an arbitrary number?
No, we haven't done that. That's a great question, and I agree with you. I think it would happen, and you probably would end up with some sort of circular pattern of dependence. It could be interesting. Thanks.
So, what resources do you imagine will be exhausted very most effectively?
In this the replication machinery, we must use resources within a cell to generate.
Or they're stuck on the other cells?
Yeah, be as,
amino acid
right.
stuff.
So you mean it's like going,
But it's replicating in itself.
And we can take it out. I'm just wondering if that's really as hard and fast.
yeah, actually, as far as I know, and then correct me if I'm wrong, but this theoretical maximum hasn't been measured in the lab. So it's something that's not really clear as to reason. Yeah, that's
There's actually a mutant that doesn't lyse that you could use
Right.
to explore that.
right.
Okay, better.
Yeah.
We tried that, we tried that, but she was just fitting a little into But yeah, in the model, you can actually change that. Any relationship between your side.
Other questions?
Can I, just one other comment maybe? Besides that, it's really cool. I just, maybe Holly knows this, I think lambda in E. coli is tail fiberless in many cases, and
That is correct.
yeah, it may have been selected so because of larger plaque formation. Almost surely there's an absorption effect of that. So there's sort of a nice case study with lambda, I think, here for this absorption issue. Maybe Holly can fill in the details of that story, but it seems like it's something relevant.
Maybe Pavitra can go into the details.
Yeah, so Romain Gillet and Iman Wang did a bunch of studies in lambda where they basically made mutants with differing number of tail fibers, and they found something similar to what we thought, which is that through being a worse absorber gave you bigger plaques, and something similar, except that they did it by mutating, generating mutants.
What was the name of the reference there?
It's Gillet et al. 2009 and 2011 with Inan Wang's lab.
Great, thanks.
Frog with no legs.
Other questions? Are there other questions?